Okay, here's lesson number 8, membrane transport. This is subtopic 1.4, and it's a pretty short list. We're talking about how particles move across membranes by different processes, including simple diffusion, facilitated diffusion, osmosis, and active transport. Look at membrane fluidity and how it allows for endocytosis and exocytosis, and vesicles are examples of that, moving things around the cells. And then we have applications. So talking about sodium potassium pumps, um, for active transport and potassium channels for facilitated diffusion. They'll come up in topic 6 that I'm going to talk about neurons, as well as looking at applications of bathing solutions in the right osmolarity. Then we're going to prepare for practical 2, which is your next practical lab about you use potatoes and see how putting different kinds of solution concentrations affects the um, osmolarity of the potato and how sugars and water and or salt move in and out of the cells based on osmolarity. So here are RAS style targets. I can analyze the transport of different particles across cell membranes, and I can use context clues to describe forms of membrane transport. Okay, so first things first, you'll be doing your little lab either today, you may have already done it, or you'll do it later. I'm not sure when it's going to happen because I'm not there. But this little lab, Ms. Couture has it set up for you guys. You're going to discuss the importance of surface area to volume ratio of cells. We talked about this at the very beginning in topic 1 in 1.1, and it's coming back here now as well. And this is, of course, directly connected to membrane transport, right? So we talked about the importance of membrane transport for moving into the cell and controlling processes, and the size of the surface area is therefore the size of the membrane of a cell. So this is why we want to have a relatively small, a large surface area compared to the small internal volume of the cell. So to better understand and visualize this, you're going to do a mini lab during class. You'll be given 3 different agar cubes. I want you to take a ruler and measure them in your notebook, and then place each of them inside some vinegar, okay, and then set a timer. After about 20-30 minutes, compare the rate of diffusion of each cell. So comparing the surface area and how quickly things move into the cell to get an idea of how membrane transport has an effect. Before you start it, or when you put the cells or the agar cubes into the vinegar, please take a minute and write a hypothesis. Which one you think will be faster and why? Okay. So pause the video and get that set up, or pause the video and find out when you're going to do it if it's not happening today. I know Ms. Couture has ready to go for you guys. That's a really good visualization and understanding of the concepts here. Let's put different kinds of transport in cells. So the concentration gradient is a super important phenomenon that's going to determine the active or passive transport of the molecules across a cell. Okay? So the concentration gradient is going to go carrying the molecules with or against it. So the gradient is looking, if you look at here, here's the concentration gradient. We have a high concentration on one side, a low concentration on the other side, and the gradient is always moving from high to low. It's the idea that all the systems want to reach an equilibrium, reach a balance, where the amount of these different solutes, different molecules are going to be the same on the inside and outside of the membrane. That's the goal. So to do so, we're going to move things across the membrane. That's a natural flow. It's from high to low. Okay? So if it's going with the concentration gradient, the same direction, you can see here the blue molecules, there's 3 of them out here, only 1 inside. That's going to be with the gradient. Yellow molecules as well, there's 3 here, 1 inside, so with the gradient, or the little red ones also with the gradient. They're going to go through directly without requiring any energy because they're following with the gradient. Now, when we're going against the gradient and we're forcing molecules to go from low concentration to high, like here we're taking that single green square and moving it out, going in opposite direction of the gradient, that costs us energy in the form of ATP. So that is then called active transport. So active transport is anything that requires ATP, and passive transport can have a variety of forms, but in general they all require no energy required because they're going with the concentration gradient. The thing to keep in mind here is the size and polarity and those kind of properties of the molecule will also help determine if its simple diffusion versus facilitated diffusion are being needed or not. So if it's simple diffusion, a molecule is just small and polar, it can go directly through the membrane. Okay, we may have channel mediated or carrier mediated diffusion, where it's going with the gradient, but it can't pass the membrane. Maybe it's just too big a molecule. Maybe it has the wrong polarity. It can't go through the membrane by itself. It has to have a protein channel in the membrane to allow it to pass through. Okay, so those are two options as well. But again, because it's with the concentration gradient, it's not going to cost us any energy. Okay. All right, let's talk about the way water moves in cells. Now, sometimes, no matter what protein channels you have, a active or passive transport, a molecule just cannot move in or out of a cell. It might be way too big or cause an issue. So instead, what's going to happen is water itself, which is very easy inside and outside of cells, across membranes, it's going to diffuse instead. So it will move across the membrane, trying to equalize the concentration of that solute that can't move. So, for example, cellulose is a very large molecule. We'll talk more about it later, but it's very, very large, and it's very hard for it to be transported across membranes. Sometimes it can't even be transported at all across membranes. So what happens is instead you might have water coming into the cell to try and diffuse it and make this happen. So if we're in a solution where there is hypotonic, which means, so hypo means low, which is a low concentration of solutes inside the solution. What's going to happen is the water is going to want to move inside where it's higher concentration inside. So because we have a lot of solute, whenever it happens to be inside the cell, the water is going to actually want to move into the cell. Now, in an animal cell, because it doesn't have a cell membrane, it doesn't have a cell wall, that can actually cause cell to lyse or burst. If too much water comes in, the cell can handle the pressure and it explodes. Okay, so we have a large hypotonic, low solute concentration in the solution in the environment around it. In a plant cell, that's how plants like to be. It helps them become very turgid or very kind of structured and have like strong support, which allows them to function really well and then be able to carry. They have it for to hold up hold up the cells, hold up the leaves. If you've seen a plant that needs some water and the leaves are kind of droopy, that's because they're lacking water to help hold them turgid. So that large vacuole we talked about in plant cells, that takes in the water and helps hold them very structured and erect. In an isotonic solution, iso means the same as, so inside and outside solution and into the cell are exactly the same. This is the ideal situation for an animal cell. Water comes in and out, everything's equal, the same concentration of solutes in and out. In a plant cell, that's going to cause water in and out, and it's actually not going to have quite enough water for it, so the structure will be a little bit softer and it'll become flaccid and not as structured in support. So the droopy leaves kind of situation. In a hypertonic, now what's happening is the environment around has a really high amount of solutes, which causes the water to want to move out to be able to dilute those solutes. When the water moves out in both animal cells and plants, that's not ideal, and the animal cell will shrivel up. The plant cell becomes plasmolized. It can't shrivel because of the cell wall, but it starts to lose kind of its cytoplasm and has lots of problems. So we have all these issues happening based on the solution solute concentration. The way water will move in and out of the membrane to match that solute concentration. Let's talk about some different kinds of molecules here and how they can move. So in passive transport, we're talking about both size and charge or polarity of a molecule that's going to allow it to move across membranes. Okay, so which kind of molecules do you think can diffuse across membranes the most easily? Big, small, polar, nonpolar. Take a minute, talk to your desk partner, pause the video, really think about this. Well, the answer is the best, easiest ones to move across membranes are small molecules and ones that are uncharged or nonpolar. They can move through because the majority of the membrane is in fact nonpolar. All those phospholipid tails are nonpolar. It's easier for the cell to move through it in that way. The bigger molecules and or charged molecules that are polar have a harder time passing the membrane. Oftentimes they require protein channels or some sort of support to get through that membrane. Okay, so here are examples of small and nonpolar: nitrogen gas, oxygen, carbon dioxide. Small and polar or charged, we have salts and different kinds of ions like sodium, chlorine, potassium. Big and nonpolar, like glucose and sucrose, these kinds of sugars, they're nonpolar, which is good, but they're big, which is not as easy to move through. They would require a membrane channel of some sort. And then we have big and polar cells as well, like water, which has its dipoles. We'll talk about more of those in topic 2, like glycerol. These kind of have a charge molecule as well. And with an OH group, anyone taking chemistry, if it has an alcohol or a hydroxyl group on it, that's going to make it move across much more easily. So harder because it's charged. And the majority of the inside membrane is lipids, are those lipid tails, which are nonpolar and hydrophobic. Okay, let's talk about active transport. So the example of this I want you to know is a sodium-potassium pump. And this is actually really good because we'll talk about this later in topic 6, talking about neurons. So it's nice that we're learning the same concept twice to help you a little bit less memorize. So I already told you the answer to this. Where is sodium-potassium pump located? It's a topic 6 connection, which we haven't done yet. And the answer is in neurons. In the axon of neurons is where you can find the sodium-potassium pump. It's along the axon of neurons. And the way it's going to work is when ATP loses a phosphate, so ATP is adenosine triphosphate, tri of course means 3, so it's adenosine with 3 phosphates attached. When it loses one, it becomes ADP, or adenosine diphosphate, and of course di means 2. So this is going to break energy from that broken bond. That energy is released. That energy can then be used to pump the sodiums and potassiums across the membrane. Now, if we look here, this pump is actually quite efficient. And when we look at this beginning here, we're starting with high sodium, low potassium outside the cell, and low sodium, high potassium inside the cell. And because we're going against the gradient, moving the sodium out, even though it's higher concentration outside, moving potassium in, even though it's lower concentration outside, that's going to require energy. But the cell is smart, and it's only going to take 1 ATP to move 5 ions, which is pretty impressive. Okay? So we're going to first move 3 sodiums out, get our phosphate, and that phosphate comes off to move 2 potassiums in. Okay? And reset the whole thing to go again. So 3 sodiums out, 2 potassiums in every single time. Let's look at this again. So once you take a minute, talk to your partner and do a tell me more here. If you don't remember what tell me more is, you need to sit with the partner and one person describes what's happening in the picture here and explains it, or the other person says, oh, tell me more, with a very interested and fascinated face and tone of voice. Really play it up, guys. Ham it up. Have some fun. Get them really thinking about, oh, this is so fascinating. Tell me more about the sodium-potassium pump. Once one person's gone through everything they can possibly say and push yourselves, give yourself a few minutes to really try and do it quite thoroughly, then you switch. The other person does the describing, and the other person says, tell me more. It's a really good tool to help you guys using language, talking about it, and just spending more time on a rather complicated process to think about. Okay, let's talk about endocytosis. So endo, we know, means inside. So endocytosis and cyto is the cell. So endocytosis is the process of bringing things into the cell. So the membrane structure is a huge part of what allows this process to happen. So take a minute and think about, look at the pictures here. How does membrane structure facilitate endocytosis? Pause the video, talk to a partner. Well, if the membranes are made as possible of bilayers, it allows them to want to keep the nonpolar insides together and the polar outsides towards the water part of it. As they pinch in, it's going to naturally want, when a little gap is formed, it's going to want to close that gap. If it breaks, it's going to instantly form a little loop, and that loop is called a little vesicle. Okay? So the flexibility of the membrane, the nonpolar inside, polar outside, that component causes it to do endocytosis in this particular way. Now, once the vesicle is formed, we have that little pinch to bring something into the cell. So these blue molecules are being brought into the cell through endocytosis. What causes it to pinch back together and reform the membrane? Well, it's all about those phospholipids, those magic phospholipids. They're hydrophobic and hydrophobic properties. The hydrophobic part of them is going to want to stay away from the water and form together and protect it from the hydrophilic part on the outside and inside. In the human body, this happens, we'll talk about it more in topic 6 later on, in digestion, when we're trying to take in certain molecules. It also happens in back in the neurons in trying to take in neurotransmitters as well. Okay, so the manage your neurotransmitters crossing the synapse between neurons. This happens through endocytosis. Okay, now the opposite of endocytosis, can you guess what that's called? You got it, exocytosis. So exo means outside, cyto is a cell, and sys is a process. So process of moving things outside the cell. And this happens a lot in relation to the Golgi apparatus. So once we're making new proteins um, in the endoplasmic reticulum, think back to 1.2 and talking about all those organelles, they're going to enter the cis side of the Golgi for packaging. And they'll be packaged there into a vesicle. Cis means this side of, or like the same side of, into a vesicle. And then that protein is going to move through the Golgi, through the whole process of moving through the Golgi organelle. It's going to then get modified in whatever ways it has to do with folding. We'll talk about protein modifications later on in topic 2, and exits through the trans or other side of the Golgi in a vesicle. So vesicles are transport little loops that are going to bring things out. Now, the vesicle can then fuse with the membrane. The same way it was made to come in, it can fuse to let things out. Because the phosphodilator is the same in the vesicle as a cell membrane, they can fuse together, and that can allow the contents out of the cell. Could be a signal to the cell, could be a waste product, could be a toxin. There's something being released out of the cell through the fusing of the vesicle membrane with the cell membrane. Okay? So endo and exocytosis are opposite processes that both rely on the phospholipid bilayer structure and polarity to cause it to create that particular pattern. Okay, here are your key ideas. So pause the video, write these down. If you have questions, please ask. I know you have a new substitute teacher who's a biologist, so please ask her some questions and make sure you understand these concepts. Okay, practice time. This sheet is on Golgi. It's cell membrane transport flowchart. Take a minute, find it. Maybe we can get it printed for you if we have it in a binder somewhere, and then fill it out. It's a great review tool to practice and check for understanding for what we've done so far in today's lesson. So take some time to do that. I'm going to change the slide to the answer, but only for a really quick minute so you can pause and check the answers later, because I want you to work through it on your own and not just copy the answers. It's about practicing, checking for understanding, reinforcing this knowledge so you have it, turning it into part of your long-term memory. So here's the answer, check it later, moving on. Okay, if you haven't already, it's time to check your data for your agar cubes. So we're moving from a cell. If it's already been more than 20-30 minutes, then please hopefully already stop them and see how far the agar has moved. And write a little conclusion to yourself. What happened? What does it mean? What does it tell us about transportation in cells? Okay, on to the next thing for today. And this is kind of our last concept for today. It's talking about osmosis and osmolarity as you prepare for practical 2. So you can see here looking at the idea of what osmosis is and what osmolarity is. So first things first, what is osmosis? Well, osmosis, you should hopefully remember from like middle school science, hopefully. If you don't remember, it's okay, I'll remind you now. Osmosis is movement of water across a semi-permeable membrane. And this is in response to trying to maintain the concentration gradient. So you see what's happening here is we have the molecules of solute, let's say when it's sugar, on each side of this tube. It's higher concentration on this side. And if for some reason the sugar cannot move, the water will move instead. So water will move to this side of the tube to try and make the equal concentration between water and sugar on both sides of the membrane. That's your membrane here. See water molecules moving across because the sugars are too big to fit the membrane. This is what you're going to be working on with your lab. Now, we need to differentiate between regular diffusion and osmosis. Why is that? Well, the number one reason is simply because osmosis only happens when the solute can't move across membrane, but water can. If a solute can move across membrane, then to make the concentration equal on both sides, the solute would just move. So small and nonpolar things can move very easily, and they're going to do that all the time. It's more often we're dealing with larger molecules or polar molecules that require a hard time across membrane that we deal with water moving instead. Okay, can you define solute and solvent? These are terms you would have learned in chemistry, probably in grade 9, grade 10. So the solute is a thing that is being dissolved in a liquid of some sort. It could be, for example, this example, the solute is sugar, and the solvent is the liquid in which you're dissolving it. In this case, the solvent is water. But we can use solvent. It could be alcohol. It could be an ether. It could be lots of different things. Very often we use water in biology. It's a universal solvent. But the solutes can differ. You'll be choosing to use either salt or sugar as your solute for your practical 2, and the solvent will be water. You could use distilled water, but you don't need to use distilled water. It won't make that big of a difference for that lab. So if you analyze the movement of water and movement of solutes, you can estimate what's called the total concentration of the solution, total solute concentration, and that is the osmolarity. So osmolarity is what is the actual concentration of that particular solute in the solvent overall. And that's what practical 2 is all about. Okay, so let's talk about the application of this. So again, reviewing iso, hyper, and hypo. What do those mean? So iso is the same as or equal. Hyper is more than normal. Hypo is less than normal. So using those prefixes and these diagrams, oops, sorry guys. Go ahead and define what those mean. Isotonic, hypertonic, hypotonic. So hopefully you get the idea that in an isotonic solution, the solute concentration or the osmolarity is the same on both sides of the membrane. In a hypotonic solution, we have a low solute concentration in the solution and therefore higher inside the cell. And in hypertonic solution, you have a low solute concentration inside the cell, but the hypertonic is the cell environment, has a high concentration. So you're going to be trying to find, the best way to know the osmolarity of a solute is if you can find the isotonic solution. If you make a solution that's isotonic and nothing is changing in the cell, nothing's moving in and out because it's all equal, then that means you have found the osmolarity point. The osmolarity, wow, of that solution. Okay, let's go to application here for medicine. And yes, that gift, if you can't tell, those are lungs that are still breathing for lung transplant. So when organs are donated and they're removed from an individual to be then transferred to the second individual, they have to be stored in the hours in between being moved between people in an isotonic solution. So we have to make sure the osmolarity of the solution they're being stored in is exactly the right solute concentration. Why is the case? Well, we want to keep the organs in the best possible shape. So if someone who's been waiting for lung transplant because they have some sort of crazy cancer or emphysema or something, and they need to get a transplant, for them, those are going to be healthy and in the best shape possible. If we store it in the wrong solution, then water will move in and out in a bad way. Okay? So if we stored it in a hypotonic solution, what would happen? Remember, hypotonic means low solute concentration in the solution. And means the water is going to all go into the cells, which means that animal cell, it would burst, which is horrible. Have an organ to save somebody's life and it explodes. That's horrible. Hypertonic solution, what would happen? You have a high solute concentration in the environment. Well, it's going to dry up because we're mixing solutions that way. Why does this keep moving too fast? Okay, so in hypertonic, it will dry up. These bags here show the kind of storage solutions you have. They have different osmolarities for storage of molecule of different kinds of tissues that are the right osmolarity to not mess with the water moving in and out. Okay. Here are your key ideas. Pause the video, write them down, and then I'm going to introduce practical 2. Okay, so practical 2, we're working on next class, and you're going to, the assessment IB is estimation of osmolarity in tissues by bathing samples in hypotonic and hypertonic solutions. So your sample you're going to use are potatoes, and you're going to use a cork board to cut them into the same size. Ms. Couture will help you with this lab and with the technique to use. And you're going to estimate the osmolarity of potatoes. If you can manage to create a solution that is isotonic, then you then know the osmolarity of potatoes. If the potatoes can be soaked in that solution and nothing will change in the potato, then you're good to go. Think about how you're going to measure the movement of water and or a solute, but you could choose either salt or sucrose as your solute. So what will you do to measure the potatoes before and after to see how they change? And I know you guys can probably Google this. Try to figure it out on your own logically first. Okay, you're going to soak potatoes in different solutions of salt water or sugar water and see what happens over several hours or even up to 24 hours. Think of different kinds of solutions. You need to actually calculate the molarity. Look at your grade 10 notes or Google molarity calculations if you don't remember how to do that. Molarity is moles over liters. So if you don't remember how to do moles using molar mass, chemistry come back at you. Maybe people who are taking IB chemistry can help you to do that. So do that before the presentation. So remembering, if we make a hypotonic solution, the water is going to move inside. If we make an isotonic solution, it'll move in and out in equal measure and nothing will change with your potato. If you make a hypertonic solution, the solute will move outside and it will shrink. So think about what you're going to do for that and prepare for practical 2 as your homework. You'll find out from your teacher when the lab will be, and yeah, the last lesson before the test, guys. Second unit, almost done. Anyway, I hope you guys are feeling okay and caught up on everything and enjoy your long weekend.